Get started. Thank you everyone for joining us today. I'm Stacy Heilman. I'm the Associate Vice Chair for Research in Pediatrics. I'm also a member of the ACME POCT, and I'm delighted to welcome you here today. Today is actually, before we get started, it's a little bit of a sad day in our ACME POCT sphere. And I just wanted to take a moment to recognize a really important member of our ACME POCT community and ask our fearless leader, Wilbur Lamb, to say a few words about Oliver Brand.
Yes, thank you, Stacy, and thank you for everyone for joining us. So a few weeks ago, we lost not only one of our principal investigators of our point of care technology center here in Atlanta, but also a very close colleague and a very close friend. Dr. Oliver Brand was a pioneer in many ways. He was a pioneer in the field of what's called MEMS, micro electromechanical systems. He was a pioneer in biosensors. And in the last 10 years, he's become a real pioneer in becoming a leader in the nanotechnology field. So he was director of the Institute for Electronics and Nanotechnology. And, if we think back, without him, there would not have been this coming together of physicians and clinicians and microsystems engineers here in Atlanta about 7-8 years ago. And without him, that would not have formed the nidus of a center under his roof within the Institute of Electronics and Nanotechnology. And without him, that center would not have become the NIH-funded center that we are now. So therefore, without him, we would actually not be here today to give these interesting and interdisciplinary talks that Oliver really was a big advocate of. So thank you.
Thank you, Wilbur. I think that Dr. Brand certainly epitomized the gentleman and the scholar both, and he will be very much missed, but his mark is left forevermore. So with that said, we'll go ahead and segue into today's program. So I did want to give a shout out to our sponsors, including the ACME POCT, as well as the Department of Biomedical Engineering joint between Georgia Tech and Emory. ACME POCT is funded by the NIH through the POCTERN network. Specifically, the ACME POCT is, that stands for Atlanta Center for Microsystems Engineer Point of Care Technologies. Our website is up here. It's really focused on the development and translation of these types of technologies, including MEMS-based sensors, microfluidics, and smartphone-based systems. Taking this a little bit step further, I just wanted to explain the ecosystem of the ACME POCT. And here in the light blue and peach, it really describes the technology development core, which is sort of the engineering-based and the mechanics behind the device development, as well as the clinical translational validation core, which not only makes sure that devices are developed with a specific clinical need in mind, but also then we have the venue and the resources to help test these devices to make sure that they're working as intended. In the light green here is the core 4 that I mentioned that I'm a member of, along with Eric Nell and Morgan Greenleaf and David Koo, which is our technology training and dissemination core. And our goal is really to take what's developed and the lessons learned in the blue and the peach areas and disseminate them out to the engineers and the scientists and the entrepreneurs, presumably all of you on the call. So that's what really GLUE is meant to do, and that's why we're here today. So with that, I'm going to now introduce our speakers. We have a little bit of a provocative title, the 50 Shades of FDA, where we're going to be doing a case study and potentially some role playing on highlighting the regulatory perspectives of a new diagnostic. And up for the challenge are two of our wonderful team members, Cathy Cambria and Erika Tyburski. So first, Cathy, I'm going to describe that she has over 30 years of experience in regulatory compliance and quality systems management. She has successfully run her own consulting company for over 20 years. Through extensive experience and continued contact with different branches of FDA, Ms. Cambria has completed multiple filings for not only Class 1, but 2 and 3 at 510K submissions and pre-market approval supplements and real-time reviews to the FDA. So tons of FDA experience. Erika Tyburski comes from a slightly different perspective, as she has 10 years of medical device experience, specifically bridging the gap between academia and industry. As evidence of this, she's CEO of Sanguina, a biotech company that makes smartphone apps and diagnostic for accessible healthcare and wellness, and also serves as part of the operations team for our Acme POCT. Cathy and Erika have been working together for Acme and the RADx program since inception, respectively, providing regulatory support and guidance for over 30 teams and companies. And so now, Cathy and Erika, I'm going to turn it over to you for your case study. Welcome.
Great, thank you. Let's see if we can get the share to switch. Are you seeing our screen?
Yes.
Okay, wonderful. And I'll do a sound check real quick. Hopefully you can hear me. This is Erika and
This is Cathy.
Sounds wonderful. You're off to the races.
all right, wonderful. Thank you for that introduction. And again, thank you so much for having us today. We're proud members of Acme POCT and all the wonderful things we do. And I will also miss Oliver very dearly as one of my direct supervisors. So thank you for that introduction. Let's see if I can shift forward. Okay, so we are here to talk to you about all of the perspectives from both an innovator's perspective and a regulator's perspective when a new device is in development, specifically medical devices in the regulated industry. In true innovator technologist fashion, we're going to frame the topic as a problem. Having an engineering background myself, it helps me digest things. And the goal is to really frame how innovators tend to be hyper-focused on creating something new and groundbreaking and novel to drive a new value. And the balance to that perspective when you are living in the regulated industry is the regulator. So keeping in mind that anything new, innovative, is potentially uncharted territory and potentially has unknown risk. And you really have to get over these hurdles in order to make your way onto the market and truly translate. So we see this time and time again. I thought it would be good to highlight it. So from a regulatory perspective, usually with a very novel technology, that means extra design, extra validation, and risk analyses to prove the safety and efficacy to a regulatory body. So today we framed that discussion in the points of view from an innovator and from a regulatory body, and we've put it in the context in designing and validating a new SARS-CoV-2 assay. So I'm Erika in the blue on the screen. I will be playing your innovator.
And I'm Cathy, and I'll be playing the role of the regulatory body.
So let's get going.
So we all know that the world has been in a state of emergency with the pandemic, and the US has had confirmed cases in every state. And with this pandemic, the world is requiring testing and screening programs for this novel SARS-CoV-2 virus. The virus is affecting everyone in schools, work, and everyday life, and scheduling becomes an issue for all. Ultimately, more testing is needed. So to meet the demand for testing in a relatively short period of time, it drove the requirement for creation of novel tests for the detection of SARS-CoV-2. And what we saw was testing expand from molecular to antigen rapid test. And we also saw that the sample types transitioned from nasopharyngeal, which was the gold standard, to mid-turbinate, to anterior nares, and even a novel sample type, saliva. With all of these things in mind and understanding that we had a huge increase in the requirement for testing, we also had to address supply shortages. And we saw supply shortages in not only the nasal swabs, but also in the transport media and consumables that were used throughout the hospitals and the instrumentation to perform testing. So with any change, It requires design and development. So we had to address the design and development for new sample types, for new users, and also for user settings. So we're going to try and walk you through a proposed COVID assay in response to this new demand. And we have an example we'd like to go forward with.
All right, so the innovator will be on the left-hand side of your screen in green, right? So from my perspective, I'm seeing that people are sick of nasal swabs. As Cathy mentioned, there's currently a requirement for a nasopharyngeal or mid-turbinate swab type. These are more invasive than we would want. We're also seeing that it is inconvenient to drive to these testing sites. We really want home testing. And we found that the public has begun to associate some of these samples with pain and begun resisting testing. So we really think there's an option here to, or the ability to improve convenience for these tests. There are requests for using less invasive sample types, especially for repeated screening, and saliva seems to have promise as a non-invasive sample type. There are no appointments available same day at testing. So the point I have as an innovator is how convenient would that be? Testing at home during symptom onset. And as a company, a small company, we realize that home tests have potential, but they're also simple and affordable, and we can market them. So from the innovator's perspective, these are all the things driving the potential need for a new assay with a new intended use and a potential business model to get them out to people that have such high demand and willingness to pay.
So it's true that the testing sites are very, very busy with limited appointments for testing, and that the traditional lab-based testing is backed up and that we need more testing, and the delays have an effect on the individual and also the population, and that the health has caused an increased time to get results, and not only that, but also to isolate and seek treatment. So home testing may be entertained as a solution, but only if the proposed test is 1, exceedingly simple, 2, safe, and 3, the benefits outweigh the risk. So what are the risks and the benefits?
And here's where, as a innovator, I would have several years ago said, what is a benefit risk profile and what sorts of benefits are important? What sort of risks are important? So we're going to have our regulatory representative walk us through what are we really talking about here?
Right. So we're going to take a look at the benefit and the risk profile for the intended use of this new assay. And we want to focus on not only individual health benefits, but also public health benefits. So what we've done is itemized a couple of benefits that we have identified. However, this is not a totally inclusive list, but we are looking at not only patient management, but patient health, the quality of life, patient satisfaction, reducing the probability of death, aiding patient functions, and not forgetting that we want to prevent disease spread and predict future disease onset, thus providing earlier diagnosis. To try and balance this out, we really need to look at the risks. And we're going to look at risks from a medical device reportable. So we're going to rate the risk based on whether or not it will cause death, serious medical injury, serious malfunction, require surgical intervention, or causing disability or permanent harm. So what we found was when we're looking at risk, we have to, for the home use, we have to look at not only self-collection from self, but also collecting from a child or possibly someone that needs aid. And that we're also have to identify the risk of incorrect result interpretation and even the false assurance. False assurance that if I'm negative, I can go do whatever I want. if I'm positive, I should isolate. So what we're going to do in this complete balance of benefit versus risk, we have to make sure that the test is safe and effective, and the probability of the patients in the public health experience one or more of these benefits increases the chances that the regulatory body allows the use of a novel test. In this case, a home test by a lay user.
Makes sense. So keeping this in mind, I think we'll proceed with our plans as a company to come up with a novel test. I think it's very important to come up with easy self-collection and collecting from someone else. We want to minimize the chance of incorrect or false results. And the whole point is to get people convenient tools to allow them to isolate and take the right action faster. So I think we have a good case here. So moving on, I think I skipped one. Let's see if I can go back one. Oh, maybe not. Okay, so moving on, again, I'm on our left-hand side of the screen. As far as our design inputs, so we've identified our user needs and what those are going to translate into our design inputs. So we'd like to do a saliva sample type that I've mentioned. We want it to be an antigen-powered test so that it is rapid and can be done at home simply. The goal is to have 15 minutes or less development time. And the idea is to use a lateral flow assay similar to others on the market that people are familiar with a simple visual readout of yes, no, there's a line, or there isn't. So in our simple workflow, we'd like to have someone swab their mouth with a sponge swab, mix that swab with some buffer, transfer drops to the sample area on the strip, and wait and read after 15 minutes. Very simple, easy enough.
So it does sound simple and easy enough, but we do have to recognize that saliva is not a traditional sample type. And with it, there is lots of variability. So when we talk about saliva and its collection, I need you to define what you're actually collecting. Is it a person spitting? Is it a person drooling? Is it a sponge collection in the mouth? Or is it something deeper such as sputum?
Oh, so we're going to go with the sponge option. So it'll be a swab that's swiped around the mouth and it will be collecting into a sponge as we go.
Okay, so a few more questions. When you're collecting your saliva, are there any time constraints or recommendations? Does someone have to wait, say 30 or 60 minutes after having food, drink, brushing one's teeth, using mouthwash, smoking, etc.?
Ooh, those are great questions, and I should probably test those. So would this fall into flex testing in order to determine our labeling and instruction?
Well, I'm sure there's flex testing associated with it, but the one that I think I'm going to focus on now is that there are many potentially interfering substances, and the situation to test and to characterize has to be defined. So we have seen that there have not been a lot of authorizations for saliva test, and that really is based on our understanding that there is variability in sample type, and that FDA has seen that saliva may not carry as much viral material as nasal samples. Easily debatable, because I'm sure there's people on the saliva side, but again, have data with FDA, and then you have a conversation. The other thing that we should also note is that you're looking to do an antigen test, which typically are less sensitive than molecular tests. So how is this assay going to be sensitive enough to detect SARS-CoV-2? What's your proposed intended use statement? I understand that you're looking to go over the counter. Over the counter would indicate that a lay person is doing the testing, meaning the home user, and that basically they can test at any point in time for any reason. And we also have to consider that they may be asymptomatic versus symptomatic. Having said all that, there's many studies required to characterize the robustness of the sample type and the intended use setting. So we need to look at your design process and your design outputs.
All right. So having gone through that, we've had some time to come together with our team and really develop an assay that delivers. We've come up with a proprietary antibody antigen conjugate that is more sensitive. You know, there are concerns about saliva, but ours is more sensitive. We've developed a new material for the swab. It's a sponge with large pores that's able to collect as much saliva as possible during collection in the mouth. We've also created custom external controls for the assay as it's saliva. And we've tested it in the lab with some inactivated virus, and it produces strong positive lines in 10 minutes or less on samples with extremely low viral loads. So we really feel like we have answers to the saliva limitations that have been put before us.
Understandable, but I do want you to consider some possibilities. How is the new antibody antigen conjugate prepared? And why is it more sensitive than the previous? Have you tested it? Have you documented it? And can you explain to me why it is more sensitive? Is it less stable than previous technologies? And you also indicated that you are going to do collection with a sponge. So I'd like some more information on the sponge material. Is it safe? How's it intended to be used in the mouth? And how is it going to be manufactured? So when we're looking at this sponge material, we're going to be looking at if it's safe and effective, and if the material that you're using has any existing documentation, or whether or not you need to do additional safety and efficacy testing. So if the material is not currently used in the mouth and manufactured in the same way, it's likely going to require biocompatibility testing. When we talk about the porosity, we're also looking at issues that may include things such as leaking or cross-contamination, not only in the assay, but also during sample transfer. So does it release all the sample for testing? Another point to consider is how you're making the controls and what an invalid result means. Have you looked at the control material? Have you validated that control material? And do you understand what would give you an invalid result and what it means? And another point to consider is, does this test result in false positives or false negatives? This needs to be balanced. We have innovators who are looking at sensitivity and specificity, and sometimes they're weighted equally, and in other instances, they may not need to be.
That's great insight. I think all of these points would be covered in our verification and validation testing plan, and we would certainly provide all of this material that a regulatory body would need. So let's get to testing. We proceed with our verification studies, essentially our analytical and bench studies. We characterize how the assay and control work. We have a good understanding of what the control does and what an invalid result means. We've done our analytical testing with saline and inactivated virus on the bench, using saline as our surrogate for negative matrix. So what do you think?
Well, I think it's excellent, but I am going to ask another question. Does the assay do what you state it can do? Basically, I'm asking you whether or not you performed a risk analysis and you've explored all the potential risks of the new assay as it relates not only to test performance, but also to efficacy and user safety. Risk analysis should be used to drive the studies required for the regulatory review. Studies should have quantifiable acceptance criteria to characterize if a test does what you claim it can do. And the other point I want to make is, although you may be able to use a controlled negative matrix for analytical studies, you should test using actual clinical matrix before reaching design lock for validation testing.
Those are excellent points. And we will test with clinical matrix during our clinical study. So maybe we should take a look at doing that and the other studies that are required for a regulatory submission. So as an innovator, I've done some of my homework. Here are the studies that I found that I need to do in order to prove safety and efficacy of my new assay. I am going to have a limit of detection done for analytical sensitivity. I'm going to have inclusivity testing done for analytical reactivity, cross-reactivity, microbial interference, and interfering substances for our analytical specificity. Because I'm an antigen test, I'm looking at hook effect and potentially the effect of biotin if I'm using that in my capture system. I'm doing the flex studies that I have found as suggestions in the guidance documents. I am also doing real-time shelf life studies on all of my test components in their final packaging, in final product. And I am starting my clinical study with a high sensitivity RT-PCR saliva comparator. So I have saliva versus saliva.
Okay, so I think what we can say here is that that's a good base list. However, based on the intended use and the new sample type, which would be a collection device for saliva, and this new user workflow, which is over-the-counter home use, I think there are other things that we have to take into consideration. Biocompatibility for the swab. So we're looking at the swab being placed in the mouth to collect saliva, and we want to make sure that it's safe, but we also want to make sure that it's effective. But to determine whether or not it's safe, we have to look at the swab collection device for saliva in its totality. And we're going to be looking at the materials, what comes into contact within the mouth, and whether or not it poses any issues of safety that need to be addressed. So again, guidance documents are available and ISO standards, which allow you to address biocompatibility issues. We also have to stop and look at your control characterization, right? So as part of your assay, you're going to have to ensure that the device works as it's intended to use. But we do want to make sure that the control is at a level such that you understand that even with a low amount of viral load, that you'll be able to, in fact, have a detection system. And then we also want to look at the stability of your sample. So again, we are talking about home use, a lay user, which really doesn't have a lot of experience with this type of thing. And you want to make sure that they have a sample, that they know how to use it, and that it is stable for the amount of time. We're also going to be looking at the interference substances that are specific to saliva. So in the past, there's a lot of information on nasal interference substances, but now we really need to be looking at the saliva itself. And I also want to ask you about your usability and human factors study. Have you taken a look at usability? And have you taken a look at human factors? And then we are going to be looking at flex studies, but the flex studies are going to be individual to your assay to your company's product. And again, these flex studies are going to be based on your risk analysis. So anytime that we're looking at the development of a new assay, we are always looking at the risk associated with not only the assay itself, but also the components and the raw materials that go into it. And then the last point that I wanted to make with you is that you spoke about having your clinical study to do RT-PCR saliva comparator against another saliva. And that's really not something that's acceptable. For the clinical study, the requirement is an RT-PCR high sensitivity nasal comparator, which is the gold standard, with a separate extraction step. And this has been documented by the FDA.
These are great points, and I'm really glad we talked about the comparator before proceeding with our testing. So as we proceed with our testing, and I put this in the hands of lay users, gosh, halfway through, I've noticed that 20% of my users are receiving invalid results. This is totally unexpected, and we really don't know what is going on at the moment. And on looking at some of the pictures that we're getting from our clinical sites, we're seeing that some of the test strips aren't wicking up enough of the saliva to wick through the control line at the end of the assay. So I'm thinking the strips must have failed. There's something we have to do about these strips.
Okay, so let's start with 20% invalids. That's really not acceptable. You're having a lot of failures where people cannot get the results that they want. Understanding that, you as the developer really needs to take a look at what the root cause is, and you have to perform an analysis to determine what actually happened. And in order to do this root cause analysis, you have to investigate, you have to test in a systematic way so that you're able to identify the root cause of the problem. And all of this has to be documented. And that's going to be a key for anything that we do from a regulatory body. Is we need to see everything documented. We need the data associated with it. If you didn't document it, it didn't happen.
Okay, so I need to be more systematic here. And what that means to me is I should learn as much as I can from this clinical data and really take a look at potential issues separately, compartmentalized. And if possible, let me see if I can replicate the problem in my own hands internally. Have I really captured what the problem is by replicating it myself? And what I'm going to do is potentially take a look at the amount of sample I'm transferring to the strip, and I'm going to use it with real saliva. And I'm going to identify in lab that I'm not getting the wicking that I see. And it's actually a combination of my paper choice as well as the viscosity of different sample collections. As you said, saliva can be variable. And what I'm seeing is that in clinical samples, the viscosity is changing. I realized, if we rewind, that I was testing in saline as a surrogate for clinical matrix in the lab, when really saline is tightly controlled. The viscosity is very nice and easy to work with, but I really should have changed the viscosity. And now I'm adding this to my list of potentially confounding factors. And I have to go back into redesign for my assay to make sure that we can actually accommodate the sample type we are claiming we can. So ultimately, I appreciate this help because I can then document it and study it and be able to directionally work from here. And I just want to clarify on the innovator side, and as we proceed in this presentation, that all of these hiccups that we're highlighting, although it's a completely fictitious case, have happened, and we have heard of them in a number of different assays. So please don't feel as though anyone's being targeted. It's things that we've seen throughout this pandemic. So That said, have I looked at my problem? Have I tested it systematically? I think I have. So what do I do now?
So again, once you propose the change, you perform your risk analysis. You have to approve the change, make the change, and perform verification testings and revalidate or retest clinically. Everything that you do has to be formally documented, and all of this has to be done according to your quality management system.
Okay. So I've re-verified, I've revalidated, I've documented the whole way. And what we found that after revising the amount of sample required per test and changing our strip, the assay was retested. We were able to complete our usability and human factors testing, indicating that greater than 95% of users know how to use the test, can correctly interpret results, and our invalid rate is now 2%, huge improvement from that 20 that we started with. And our clinical study results are just in, indicating that our positive percent agreement are at 95% and our negative percent agreement are at 95%. So now, do my results meet the bar?
So again, we're going to be looking at your device in its totality, and we want to understand, does the assay meet the performance criteria of other assays for the same intended use? So typically, for a binary assay, there's a trade-off between sensitivity and specificity. In the case of an OTC COVID-19 assay, it's very important to know the performance criteria to be met prior to the validation, because there are different benefit risk profiles in different use cases, which drive the different acceptance criteria. So for example, as an OTC COVID-19 assay, the NPA actually needs to be greater than or equal to 99% with a negligible risk of false positive. FDA has taken the position that across the board OTC tests are positive, they're true positives, and are being used to make decisions on isolation and treatment. Therefore, this 95% NPA actually doesn't meet the performance criteria for an OTC COVID-19 assay. I'm going to ask a few more questions. Are there any new safety risks in the home use environment? Have you identified them in your risk analysis?
I believe so. We've done a comprehensive risk analysis at each of these stages, and then having to go back and revisit it in changing the assay, did a risk analysis once more. It does seem like we will have to improve on that NPA, and we did fall to the optimization of both sensitivity and specificity, as you were talking about earlier. We really should have taken a good look at what the benchmark is and what the current thinking of the regulatory agency is with respect to optimizing these two performance criterias. But once we do get it figured out, what would be the process we do?
Well, let's just be clear that if whatever changes you have been made, you need to go back and make sure that those test criteria that you have done actually are on the final product device, right? So you may have to do some repeat testing. But let's assume that you have done all of your testing. It's now at a point where you're ready to submit your submission to FDA. And in this submission, you want to make sure that you provide FDA with all of the information so that they can make a favorable decision. So we're talking about that we're looking for a certificate of analysis for some of your raw materials and their components. We're talking about the protocols that you use to perform testing. We're talking about the raw data, any final reports, which basically summarize that raw data and take everything into consideration. And that if you have used guidances or standards for utilized in your studies, that you reference those guidance and standard documents. So FDA does have consensus standards which are recognized. They're always beneficial if you can reference them. There are other guidances which FDA has published And in the case of the emergency use authorization, we actually saw FDA published templates. All of these things are excellent references to help in your regulatory submission and to aid FDA in a favorable review.
All right, thank you. So at this point in the presentation, we're going to stop our mock case and the many hiccups and trip ups we had and want to bring it home for the audience. So what I'd like to pose is, do any of these slides sound familiar? We talked about user needs, design inputs, the design process. We talked about our design outputs as well, verification testing, and finally validation testing and getting it to a regulatory clearance. What we've actually done is highlighted the famous waterfall diagram. This is a diagram used in FDA guidance to describe a controlled development process. What we've done is highlighted in a mock case going through the example of designing, verifying, and validating a potential assay for COVID-19 to get it to a regulatory submission. And I do want to be clear about this. I want Cathy to chime in as well. This process in general can be used on an even larger scale in your companies after a regulatory submission and or your authorization or clearance is obtained.
And that's right. So we are looking at this waterfall, which is a representation of a controlled and systematic development process for a medical device. It identifies the control of design across the product life cycle, and it starts at product feasibility and continues through product termination. And that basically means from inception all the way through when you no longer manufacture or sell that product. But another key that needs to be highlighted is it requires the development team to have representation from all departments. And the departments that we're talking about, which is not all inclusive, but should factor in research and development, quality, regulatory, manufacturing, marketing, just to name a few. And that reviews and risk analyses are going to be performed at key development phases. So the phases are documented for completeness of milestones, reviewed and improved in order to advance to the next phase, understanding that it may be necessary to revisit a phase after a device failure or identification of possibly a new user, and the need to go through the phases once again.
Right. This is a very powerful diagram that we get questions about all the time. You know, what exactly does this diagram mean? So hopefully we were able to shed some light on this part of the black box. And the black box question I get all the time is, what does FDA really want? And the simplest answer is there's no prescription and there's no template that will include everything on what a regulatory body would like to see with a device. It's because they're very specific. You are making something specific to a specific user need, and the potential risks and benefits will be unique for your assay. So unfortunately, there's no golden template for every little thing. But if you follow a controlled and intentional and systematic documented process that includes continued risk analysis, and you present those risks against the potential benefits in the context of your regulatory submission, which means putting them in the context of safety and efficacy and putting them supported by data and documentation, this is really what's wanted. And it's not really a secret. It is a process, and being in the medical regulated industry, something that you should adopt for your entire company's practices. Finally, to close, we do want to keep in mind with our innovators, this is not a presentation designed to stifle innovation. We need to continue inventing to meet user needs, to make things more convenient and achieve all of those benefits we've talked about. We need to design with user needs in mind, identify risks early and often in every single stage, test methodically, control your testing so that you know at the end of the day what went wrong or what went right, in order to create safe and effective solutions. So at that point, we wanted to turn it over to the audience for questions. And if you'd like to contact us again, we're Erika and Cathy from the Acme POCT regulatory crew. We're also part of your RADx ITAP teams if you're involved with them, and we'd be happy to follow up.
Thank you, Erika. Thank you, Cathy. Just a reminder, if folks have questions, feel free to put them in the chat, or if you're feeling extra brave, you can raise your hand and we will unmute you. We will start with a question from Bob Jarris, which is, what is the FDA's benchmark for positive % and negative % agreement? And I assume, Bob, this is specific to the case laid out, because I'm sure it changes for the different assays. But what would you say to that question, Erika?
Yeah, that you're hinting at it right there at the end of your statement, Morgan. It truly does depend on the use case and the benefit risk. So as we went through for a home test, the position was made pretty early on that there's not going to be high rates of false positives for home tests, because we really wanted the regulatory agency and the public really wanted to be able to rely on them to be tools to direct self-isolation and testing. So in actuality, on home tests, you're talking about true positives that are treated as true positives and presumptive negatives, i.e. you could still be positive if you're negative, but it is extremely likely that you are positive if a positive result is coming up. And that comes back to the trade-off with a binary assay between sensitivity and specificity. And Bob, to get to your question, it truly does depend on the use case. So in a high complexity or moderate complexity environment for antigen versus molecular, there are different criteria. And in my opinion, I'll let Cathy chime in too. I think it comes down to supply and demand as well. So what is the demand for high sensitivity assays on the molecular side? What is the demand for rapid antigen tests? And fully knowing that the way antigen tests work, they likely have lower sensitivity, but there is a place for them. So when you're thinking about a new assay, it does really depend on where you're intending to place it to answer those questions on positive % agreement and negative % agreement. In general, for a home test, for an antigen home test, we have seen assays above 80% positive % agreement and above 98 to 99% negative % agreement. What do you think, Cathy? Anything to add?
No, I would agree with that. We definitely do see different criteria for molecular assays versus antigen assays. And we do see differences in labs that are basically high complexity, moderate complexity versus those that are point of care, and then ultimately the home use. So it really depends on not only the user setting, but also the user themselves.
Mm-hmm. It also depends on if there is a shortage or not. So again, if there's a certain type of assay that's associated with a certain performance, but there are no other assays available, right? And we saw this very early on in the pandemic. There is a judgment call to be made on risk and benefit. So there's also that to keep in mind, especially if we're talking about an EUA, which we didn't really create this PowerPoint to talk only about emergency use authorizations. But in that case, you're also worried about how long and how fast we can get testing out there. And those things were also weighed into the risk benefit.
Awesome. Thank you for the excellent answer. We did have a very long question from Tyler in the chat. And I don't know if you can see that directly. I don't know if I'll read the whole thing, but it essentially comes down to, with more molecular devices being available and being approved by the FDA, they are still requiring NP swabs as comparator for all of those authorizations. And do you see the FDA modifying their guidance to AN swabs or other sort of less invasive swab types as comparators?
I think we'd need more context. I mean, there are certainly assays that have used sample types other than NP to secure their authorizations. So Tyler, I would say if there's a specific use case, shoot us an email and we'll learn a little bit more about it, but we'd need probably the totality of what the ask was. Is this a de novo submission, an EUA? Is it a completely novel sample type? Because it'll also depend, right? So if you are creating a product that is very, very similar to ones on the market that, for example, an anterior nair sample, lateral flow assay for over-the-counter use, I don't know that you'd necessarily need to reflex to an NP for an authorization. However, if you were doing a sample type like breath, for example, you may have to reflex to the gold standard sample type. So it would really depend.
Got it. And he mentioned that this is raw saliva for EUA.
Mm-hmm. So in saliva, again, that was the example we walked through. So we have certainly seen a request from FDA to reflex to the gold standard sample type for saliva assays, especially if it's paired with a new type of technology.
Right. Is that because saliva is just so much less common as a sample type?
Well, common in the sense of having been put on the market as a medical device or as an emergency use authorized device, I think so. And for many of the reasons we spoke about today, in that a saliva sample type, there are many different nuances in the sample type itself. So is drool technically a different sample type than swabbing the mouth? Is forcefully spitting and producing saliva that way a different sample type based on the actual solution you get at the end of the day? So some would argue that those are actually different sample types and have different risks and different studies associated with them. So I just think there are a lot more unknowns that have not been universally characterized.
Got it. By the way, I love the use of depends. It's the classic consultant answer, the ultimate. It depends. Okay. If anyone else has questions, please put them in the chat. I have one follow-up based on what you went through in your case, which is related to a risk analysis. So when you were talking about the move from saline to saliva and that produced results that were unexpected and you needed to do a risk analysis, can you give an example of exactly how to do that?
Yeah, I think so. So in a traditional risk analysis, you are trying to identify things that could go wrong. And then you're trying to identify how likely they are to go wrong or to occur, and how severe would the outcome be if it were to come to pass. And where possible, quantifying it, which is wonderful for me as an engineer with that background. Love quantifying and rating things against one another. So as an example, you could say there's a risk that someone is going to have a high viscosity of saliva because they're sick and there's lots of mucus involved. And what's the likelihood that this could happen? Well, we saw it in our clinical study as much as 20% of the time happening. So there's a criteria, right? So maybe it's a scale of 1 to 5 on incredible probability, like it would never happen, like 0.01% chance. Maybe your next level is up to 10% chance it would happen, up to 20, and so on and so forth. And you'd be able to assign it a score on likelihood. And then you'd say, well, what's the outcome? What is the severity here? And here's where it becomes interesting. So as an invalid result, which is what we were seeing in our mock case, that's not necessarily going to cause false assurance. It may cause inconvenience and frustration with the lay user because they don't have a result, right? But perhaps that severity would have been higher if it actually produced a false result and gave the user false assurance that they were negative when actually the viscosity was interfering and it should have been a positive result. So then laying all of that out there, you'd assign your scores as best you can. And at the end of the day, multiply them or add them together, weigh them. Usually they're weighed equally, likelihood and severity. And then it has a risk score. And then as a developer, what you should be doing is understanding, how can I mitigate these risks? So you'd have a list next to it that says, well, I'm going to include instruction that you need to this is totally just an example. You need to clear your throat or something to clear any sputum out of the way and, make sure you're not getting that really viscous sample. that could be something, and you'd have a flex study to confirm that that was a suitable solution. So it could be a combination of labeling and how you design it. So again, we went back into redesign, picked a different paper, picked a different size sample to ensure that you'd have enough to wick through, even with a little higher viscosity. So that would be an example of just one line item on a risk analysis, which honestly could be hundreds of lines long by the time you get done.
Daunting.
And ultimately, what you're looking to do with your risk analysis is to not only understand those risks, understand how you can mitigate those risks such that they can be brought to what is an acceptable or tolerable level, right? So at some point in time, we understand that you'll never get it to basically no risk, but you can definitely reduce the risk by doing certain changes to the design of your assay, or even perhaps the labeling that's provided with your assay.
Got it. Great example. Last question, and this has to do with something you said at the very top, Cathy, which has to do with the assessment of safety in the context of supply. And does the FDA assessment of demand and supply occur only for a public health emergency like Mpox and COVID, or does that happen with non-EUA, so 510K or de novo clearances as well?
So I do think that FDA is going to be looking at the assay in its totality. We actually do see it highlighted because of the public health emergency, and wherever there's a need. So in that respect, as far as supply is concerned, if testing cannot be performed and there is a need out there, I do think that FDA may actually try to help address the issue.
Got it. Okay, and then one logistical thing is, will you make these slides and guidance documents available for the participants today?
Yes, we will follow up with a list of guidance documents that we spoke about on this presentation, and we'll send you the slides, Morgan.
Awesome. Thanks everyone for joining. And don't forget to fill out the survey, please.
Yes, you'll have an opportunity to tell us about today, as well as tell us about what future topics you'd like us to cover in Glue. So please do respond. We look forward to hearing from you.
Great. And thank you, Cathy. And thank you, Erika.